let me start. Actually, Jan raised an interesting question, which is really how do we define aging? And what I'm showing you here is actually it's an evolutionary biologist's definition of aging, which is really that this is simply a process, or probably a series of processes. So I think most of us would agree that there's no single aging process, but there probably aren't a million either. And those processes take a young, fit organism, an organism that is capable of both physical and mental acuity, and changes it into an organism that can really only be defined as less fit. Now, of course, nobody dies of good health. So what you die of is a disease, usually. And what I've listed here are really just a partial list of a number of the diseases that have as their component, either as a cause or a contributing factor, a very large cause and contributing factor, aging. And what we know is that what the aging process then does, whatever those aging processes are, is it makes the organism susceptible to developing these diseases. And if you're lucky enough to live a long life, you will almost certainly develop one of these diseases. Actually, you'll probably develop multiple diseases. And these comorbidities is really what causes this great decline in health span that, that most of us will face if we live a very long life. So, it doesn't matter what your age is, whether you're 16 or 60 or older than that. We should really care about aging and this large health burden that it's going to impose upon us, or is already imposing on us. And I think the next two slides illustrates specifically why we should be concerned. So this is a population structure. This is the population in the US in 1950. And if you were to go out into the wild and look at a bunch of mice or bunch of feral cats or whatever, you'd see a population structure that looks something similar to this. And that is that there are lots of young people and many fewer old people, and very, very, very few people that have extreme longevity. And so these are females and these are males, and this black line here is around 50 years of age, so sort of the midpoint of the human lifespan. Now, of course, we all know what's coming, right? Whoops, I keep going the wrong way. We all know what's coming, whoops, and that is this. So this is the projected population structure for the US in a very short period of time. This is projected for 2030. And you can see what's happened is there's this huge bulge of older people over the age of 50, and also many, many more people now at the highest stratosphere, the highest ages. So this is not such bad news. This is actually good news. You know, our species is actually, at least in the developed world, our species is living longer. So that's good news. But there is the bad news, and the bad news is that it's really crippling the economies of the developed world. So this again is slightly out of date data. This is now more than 10 years old. But what this graph shows is the cost of the last 2 years of life normalized to people who are in this age bracket, 25 to 34. So if you normalize everything here, obviously for very young people, it's not very costly. It costs about half as much for the last 2 years of life. But you can see what happens after about this age. It's actually this age, 34 to 44. The costs just escalate, and so it costs 5 times more for the last 2 years of life for somebody in their 70s than for the last 2 years of life of somebody who's in their 30s and 40s. So we are living longer, and it's pretty clear that this longevity increase that we've achieved, we as humans have achieved, is costing us. It's costing us emotionally because those last 2 years of life, as I said, are usually fraught with comorbidities, and it's costing us financially. And so the question is why? Why did this happen? And the answer, I think, is also pretty obvious. So modern biomedical science has become very good at treating certain individual diseases. And so death from those diseases is often postponed. But what we're not very good at, we still haven't conquered, is getting at the underlying causes of those diseases. That is, those basic aging processes that are driving all of those diseases. So, so if you look at these diseases of aging, the ones that I showed you on my first slide, what is clear is that they're all driven by an aging process, and that's clear because they all rise with approximately the same trajectory. So this is a hypothetical diagram of the incidence of any one of those diseases over age, and what's striking is that all of them rise with exponential kinetics, starting at about the midpoint of the lifespan. So for a human, this is 50 to 60 years of age. And of course, the calculations have been made about what would happen if we took those diseases and cured them one at a time. This was work that was pioneered by J. Olshansky. And the calculations are really quite sobering. So if you imagine this is age, and this is individual diseases, and this is the termination of life of a person, say, due to cancer. So what J. Olshansky calculated was if we did away with, if we cured cancer tomorrow, we would probably add a year or two to human lifespan. And the reason, of course, is because there's heart disease that's progressing, and if this person didn't die of cancer, they would die of heart disease. And if we were to cure cancer and heart disease, we'd still only add maybe 3 or 4 years of life to human lifespan, and that's because this person would probably die of a stroke or die of some sort of dementia. And that's because there is these basic aging processes that are driving all of these diseases. So obviously the challenge for people like us who work on the basic biology of aging is to try to understand what are these processes that drive all of these diseases and try to tackle this multi-system decline, because this is where the hope is of not only extending health span, but also dealing with multiple diseases at the same time. So it's kind of a pipe dream, and it's still not clear that we can do it, but I hope to show you some data to suggest that there might be some paradigms on which we can work, which give us some hope. So what are these basic aging processes, and what can we do about them? And of course, evolution has done a great job. Evolution has taken a mouse with a 3-year lifespan and made a human with a 100-year lifespan. 97% of our genes are identical. So evolution can do it, but we don't want to wait 100,000 years for this to happen. So let me show you one way that we've sort of thought about this problem and come up with at least one model or one process that might drive multiple age-related pathologies. And I don't want to leave you with the impression that this is the only problem. So if you think about these diseases of aging, what is really striking is how most of them are degenerative. And by degenerative, what I mean is a cell, or more likely a tissue, doesn't work as well as it should. And so tissue function or organ function declines. And then there's this disease here, cancer. And I would be very hard-pressed to call cancer a degenerative disease. I'll talk to you in a few moments about what it takes for a single cell to give rise to a lethal tumor. And this is not a loss of function. This is, if anything, a gain of function. So if you think about things this way, you think about some diseases of aging as being loss of function diseases, and some diseases of aging, most strikingly, and of course the most lethal of which is cancer, as being a gain of function disease. then you can go on this holy grail search for a basic aging process with a slightly simpler question. And that is there a common biology that links these degenerative changes with cancer? Because if there is, this might give us clues as to what this basic aging process might be. So I'm going to argue that there is a common biology between cancer and the degenerative diseases of aging, and argue that by understanding that link, we might have a handle on at least one of those basic aging processes. So let me start with cancer. You heard from Jan, and you'll hear more in this meeting about what are the drivers of cancer. And I think there's no question from many, many decades of cancer research that the primary thing that drives cancer is mutations, somatic mutations. So mutations that you acquire during your life as an adult. I also want to remind you of Jan's point just before he left, which was that mutations accumulate very early in life. We know that from those mouse models, and we even know that from some human experiments. And another point, which I think is often overlooked, is that it's not 3 or 4 or 5 or even 10 mutations that's required to develop a cancer. In fact, if you look at a relatively benign cancer like ductal carcinoma in situ of the breast. So these are cancers, they're bad to have, but they're not very aggressive. And if you do something like comparative genomic hybridization, what you find is those cancers, those relatively benign cancers, have hundreds of mutations. And now you can compare that tumor to a metastatic lesion that's going to kill a woman, and again, you find pretty much the same hundreds of mutations. And so this gave rise, I think, very early on, these kinds of comparative studies, to the idea that although there's no question mutations are needed for the development of cancer, it might not be enough. And there is something else, I think, that is important for the development of cancer, and this again is an accumulation of many decades of cell biology. And what we now know is that you also need a tissue environment that is permissive for the development of a lethal cancer. So you can have an oncogenic genome with tons of mutations, but you still need the right tissue environment in order for that mutant cell to give rise to a tumor. And I should point out, this concept goes back decades. Those of you who study developmental biology, you may remember some very classic experiments done by Beatrice Mintz, where she took a tumor, she grew it up in culture, took those cultured tumor cells, put them into a mouse or a rat. The mouse or rat developed a horrible tumor. No surprise, the mouse died. But then she could take those same tumor cells and put them into a different tissue, a different location within the animal, and nothing happened. The animal was fine. And it was the first hint that you do need a tissue environment, even in the face of a very mutant genome. And this is an example from my colleague at Lawrence Berkeley Lab, Mina Bissell. These are normal human breast cells. They're growing in a 3-dimensional culture. The red are the nuclei. The green is a protein called ecadherin, lines up between the cells. Beautifully organized, looks a lot like what would happen if you sectioned through a normal human breast. And this is breast cancer. And you can see it's a mess. The cells are not organized, the nuclei are lobulated, ecadherin is all over the place. But now, these breast cancer cells were tricked with a molecular trick, in this case it was an anti-integrin antibody, into thinking they were in a non-permissive environment for developing this horrible, disorganized structure. And the cells simply reorganize and form what look, again, like a very normal structure. So you need 2 things to make a cancer. You need mutations, and you need a tissue that is permissive for the development of cancer. I've told you that mutations, and Jan told you earlier, mutations accumulate very early in life. So everyone in this room, I guarantee you, you've got cells with mutant P53, or you've got some cells with a translocation that's potentially oncogenic. But hopefully no one in this room has cancer. The other thing is that we know that tissue structure begins to degrade also long before we really begin to see clinical disease. So you can tell a 40-year-old liver from a 14-year-old liver, but that still precedes the development of most clinically detectable cancers by at least a decade. So if it's true that we've all got mutations, we've all got our tissues degrading at an early age, the important question is, why don't we get cancer more often than we do? And the reason is because during the course of evolution, during the course of evolving organisms with renewable tissues, evolution had to solve the cancer problem from the very beginning. Otherwise, we'd be dead of cancer before we had our babies. And of course, evolution did a great job and gave us literally hundreds of genes that we now collectively call tumor suppressor genes. And what these genes do is they put into place cellular and tissue centric mechanisms that act primarily to suppress the development of cancer. And now I'm going to argue that that great feat of evolution came at a cost, and that cost is aging. So let me develop for you now the argument for why we think this is the case, that there's been this evolutionary trade-off, and then I'll show you some data to suggest that, well, we still may be wrong, but at least we're still on the right track. Okay, so let's start with tumor suppressor mechanisms. About 15 years ago, Kinsler and Vogelstein, two very prominent cancer biologists, reminded us that it would be very useful, it could be very useful for understanding the biology of cancer as by categorizing tumor suppressor mechanisms or tumor suppressor genes as coming in two basic categories. And they call these categories caretakers and gatekeepers. So caretakers are very straightforward tumor suppressors. They take care of the genome. So they protect us from cancer by suppressing the development of mutations. So these are our favorite DNA repair genes. These are genes that suppress, say, oxidative damage to the genome. And in fact, these genes can be considered longevity assurance genes. So they help assure our longevity by taking care of the genome and suppressing the development of this pathology that would ordinarily curtail longevity, namely cancer. Now, the gatekeeper tumor suppressors are actually much more complicated proteins. P53 is a quintessential gatekeeper tumor suppressor. And what these tumor suppressors do is they are integrators of cell physiology. So they receive multiple inputs of cell physiology, and then they execute large programs, usually at the level of transcription, that determine cell fate. And for the purpose of preventing cancer, the two cell fates that are crucial is cell death or apoptosis, or a terminal permanent arrest of cell proliferation, which is also termed cellular senescence. So I want to emphasize that both these cell fates are really crucial tumor suppressor mechanisms. So we have mouse models and some human mutations where cells don't undergo these cell fate decisions very well, and we know what happens to those organisms. They die an early death due to cancer. So these are good tumor suppressor mechanisms. But you can imagine now 50 or 60 years of a tissue constantly losing cells through apoptosis as a tumor suppressor mechanism. Eventually, that tissue is going to lose its progenitor cells and therefore its ability to undergo repair and regeneration, and it's going to experience atrophy, or what we call degeneration. And the same thing is true for tissues that accumulate non-dividing senescent cells. So these cells I'll show you in a moment are actually much more complicated than apoptotic cells, and eventually the tissue that accumulates these cells is going to also be unable to undergo regeneration and repair because the tissue is filling up with non-dividing cells. And then I'll show you data that these cells can also positively have detrimental effects on neighboring cells. So this is aging. This is precisely what we call an aging phenotype. And so the argument is that the price we paid for being relatively cancer-free for half our lifetime are self-fate decisions that eventually drive those phenotypes that we recognize as aging. So I'm going to focus on cell senescence because this is mostly what we work on, and develop the argument a little bit further and show you some data to suggest that we're on the right track. But just bear in mind that the argument also holds for this process of apoptosis. So for those of you not familiar with senescence, this slide summarizes many, many years of work, more than 5 decades of work from many laboratories, but it just gives you a flavor for what I mean when I say a cell is senescent. So you think of this as a self-fate decision that is possible for a cell that can divide. So we don't think things like neurons undergo senescence. These are cells that have the ability to divide, and then they become locked into a growth state that is essentially post-mitotic. So it's as though a breast epithelial cell became a neuron. You never get tumors out of mature neurons because they can't divide. We now know that there are many, many potential cancer-causing stimuli that can drive a cell to adopt this self-fate. And those stimuli range from things that cause genomic damage of the type that you heard earlier in this meeting, but they also include things like, for example, a mutation that activates an oncogene. For most cells, that activation step, that mutation, will drive a cell into senescence as a protective mechanism. And the only way most oncogenes can really transform cells efficiently is if there's been a pre-existing mutation that prevents cells from undergoing one of these self-fate decisions. And then finally, the last point I want to make is that unlike apoptotic cells, senescent cells are not dead. These cells are making protein, they're making RNA, they don't synthesize DNA, but they are metabolically quite active. And in culture, we know that they're extremely stable, so we can keep them alive for years on a culture dish. In vivo, of course, we don't really know what their dwell time is. So in the last decade or so, we've now been able to, when I say we, I mean the field collectively, has been able to identify a number of genes that are pretty good biomarkers of senescent cells. Although I want to make the disclaimer that, to my knowledge, there is no such thing as a truly senescent-specific marker. But we have now a collection of markers, and I think most people are fairly comfortable that when you have a certain number of markers, you can probably say that that cell has become senescent. And that's enabled people to go in and look at tissues in both human, rodent, and non-human primate tissues and ask, number one, do the cells accumulate with age? And the answer is yes, they do. And number two, are they present at sites of those age-related pathologies that I showed you on my first slide? And the answer is also yes. And what's interesting is you find these cells not only at degenerative pathology, so for example, arthritic joints, but you also find them at sites of those hyperproliferative pathologies, for example, precancerous lesions. So the cells are at the right place and the right time to be driving age-related disease and age-related dysfunction. The question is, do they really do that? And if so, how? What's the mechanism? So I'm going to try to answer the second part of the question first, and then speak a little bit about the first part. So in order to understand how a senescent cell might drive an aging phenotype, it's important to understand that this senescence response really involves a total reprogramming of the pattern of gene expression. And when you examine the kinds of genes that change when the cells become senescent, one thing that is really striking is that a number of genes are turned on, upregulated, usually at the level of transcription, And those genes encode proteins that can act outside the cell, that is, secreted proteins. And so we've termed this the senescence-associated secretory phenotype, and this is a picture of what it looks like. So what you're looking at is the results of an antibody array. So these are little filters. This particular array has 120 antibodies dotted on it. And this work was pioneered by Jean-Philippe Coppet when he was in the lab. And what he did is these are 5 different types of human fibroblasts from fetal all the way to adult, and they're from skin, breast, and lung. And so it doesn't matter about the body site. He's made them senescent in culture, and actually it doesn't matter how he makes them senescent. It can be done, we like to use ionizing radiation because it's synchronous and doesn't require metabolism, but it really doesn't matter. And he's compared them to their quiescent or non-dividing counterparts. So then he collects the conditioned medium, he puts them on the arrays, the computer takes signals from all of the antibodies, and then we ask the computer to average the signals and show everything that's above average as yellow and everything that's below average as blue. And so this just shows you that these cells are secreting lots of things. Now, this is not a general upregulation of secretion, because these are about 40 different cytokines here, and I told you that the array has 120 antibodies. So there are lots of things so the array goes way out here. Lots of things don't change, but there's these 40 or so cytokines that change very dramatically, upregulated very dramatically. So if we look at the biological activities of all of those things that the cells are secreting, one thing that is really striking about those proteins is that they are predicted to be pro-inflammatory. And that is the most conserved feature of this secretory phenotype that we have found. And when I say conserved, I mean both between mice and humans, but also between different cell types. So I showed you data for fibroblasts, but we get very similar data with epithelial cells, prostate and breast of what we looked at. Endothelial cells, very recently, we've shown that astrocytes show a very similar pattern of this high level of pro-inflammatory cytokines that are being secreted. Well, the cells also secrete growth factors. They secrete things that will remodel the blood supply. And they also secrete things that talk to the immune system, but also enable the cells to evade immune detection. And I won't have time to talk about this, but if anyone is interested, I'm happy to tell them about how the cells talk to and evade the immune system. So this is exciting because if you think about all of those diseases that I put on that first slide, all of them have as either a cause or a major contributor inflammation. This is the driver of a large number of age-related pathologies. I'm sure you're going to hear more about that from Claudio. So if you think about now tissues accumulating cells, senescent cells that can't divide, but now in addition to their being unable to divide, they're secreting pro-inflammatory cytokines, you can imagine that these cells are one aspect of this so-called inflammaging, as Claudio coined the term, the driver of age-related inflammation. And as you'll hear later, I'm sure from Claudio, aged tissues are often said to be undergoing sterile inflammation, and that's because, unlike an acute inflammatory response, the signs of inflammation are there, but there seems to be no evidence of a true invading pathogen. So I want to say one other thing about inflammation. So pathologists have a way of deciding whether a tissue is inflamed, and that's typically by looking for the infiltration of leukocytes, damaging leukocytes. So these leukocytes, in an acute inflammatory response, those damaging leukocytes are primarily there to kill an invading pathogen. So they're making hydrogen peroxide and hypochlorite and nitric oxide, all these toxic molecules that could, in theory, destroy the local tissue. In chronic inflammation, the leukocytes are there. There's no sign of an invading pathogen, and they're there at a fairly low level. And that's typically how pathologists decide whether a tissue is inflamed or not. Of course, those leukocytes were called to the tissue by cytokines that are attracting them, the cytokines that senescent cells make, for example. I want to point out, though, that those cytokines can have effects on tissues that have nothing to do with the immune system. So, for example, interleukin-6, IL-6, classic pro-inflammatory cytokine, IL-6 can cause certain epithelial cells to undergo what's been called an epithelial to mesenchyme transition. So to put that in context, epithelial cells are designed to talk to each other. They tend to grow and organize tightly, and there's communication between the cells. Mesenchymal cells are lone rangers. They're like fibroblasts. They break away. They're not necessarily in contact with each other. They move around. So when an epithelial cell now breaks from its neighbors and becomes more mesenchymal-like, it now is compromising the ability of the epithelial sheet to function. And for those of you who are familiar with the cancer field, you know that when a pre-malignant epithelial cell undergoes this mesenchyme transition, that's a very deadly step in the development of serious cancer, because that enables the pre-malignant cell now to begin to invade and eventually metastasize. So it's important to remember that the presence of cells that are secreting these cytokines can have two effects. They can call destructive leukocytes to the site of the tissue, and those cytokines themselves can disrupt normal tissue structure and function. So over the years, we've studied the secretory phenotype, and I'm going to show you 3 examples of things that we know it can do. And then I'll show you sort of the model that we're still working on. So the first example is an example of senescent cells causing a normal tissue structure and function to decline. And this is an example from the mammary glands. So what you're looking at here is a 3-dimensional culture of mammary epithelial cells. These are from a mouse. They were actually isolated from a mid-pregnant mouse. And if we put those cells in a 3-dimensional culture, they organize into alveoli. And then if we add lactogenic hormones to those cultures, they actually start to produce milk. So they're differentiated. And that's what the red here is now. This is staining for beta-casein. If we embed in these cultures non-senescent fibroblasts, mouse or human, it doesn't matter, we still get these lovely alveoli, lots of milk production. But if we embed in them senescent fibroblasts, you can see that milk production falls, and of course the alveoli now are beginning to become misshapen. So the presence of these senescent cells now are destroying the normal function and structure that these cells would normally form. And we now know that a lot of this is due to the matrix metalloproteinase 3 that senescent cells make. Here's another example. These are human breast epithelial cells growing in a 2-dimensional culture. Conditioned medium from non-senescent fibroblasts keeps these cells growing as they love to do, touching each other and growing in tight colonies. But if we now add conditioned medium from senescent fibroblasts, you can see what happened is the cells are now pulling apart and they're beginning to migrate. So this is that epithelial to mesenchyme transition that I told you about. And it's due primarily to these 3 cytokines that the senescent cells are producing. So we can show all this by using blocking antibodies. And then finally, I'll show you a rather shocking example from cancer. So here are pre-malignant mouse epithelial cells growing in a mouse, nude mouse. These are pre-malignant, so they're not normal, but they don't have enough mutations to form a tumor, and they don't over the course of 6 months or so. And if we co-inject them with pre-senescent fibroblasts, they still don't form tumors, but in the presence of senescent fibroblasts, these cells will convert to full-blown tumorigenicity. And this is a similar example. These are human breast cancer cells growing for 6 weeks in a nude mouse. We excise the tumors. This is what they look like. But if these were co-injected with senescent fibroblasts, you can see the tumors are larger, and these tumors are also much better vascularized than these tumors. So these data argue that a senescent cell can disrupt normal tissue structure and function and even drive late life cancer. And I just want to point out a paper that was published by my colleagues at the Mayo Clinic a few months ago. They created a transgenic mouse where it was possible to kill senescent cells by administering a drug. We've made a similar mouse, and I'll talk about that mouse tomorrow. But in their paper, what they did was they were able to selectively kill senescent cells in vivo in the mouse. They didn't do it with natural aging. They crossed those mouse to a premature aging mouse model. And although clearing senescent cells didn't extend the lifespan of those mice died of a cardiomyopathy, the mice also developed sarcopenia, osteoporosis, and cataracts. And by eliminating senescent cells, they could eliminate the sarcopenia, the osteoporosis, and the cataracts. So again, this argues that the presence of these cells can drive age-related pathology. So here's the model. Here would be a tissue with an epithelial layer in contact with the basement membrane and the stroma, and this is the unit of function within an organism. And with age, cells become senescent for whatever reason. For ionizing radiation in the atmosphere, from oxidative stress due to our own mitochondria, mutations that turn on oncogenes, it doesn't matter. The cells become senescent. They begin to secrete these molecules that will now destroy the local tissue structure and therefore function. And if there happens to be a pre-malignant cell nearby, which we all have, then that pre-malignant cell can now be unleashed from the tissue microenvironment. It's put now into a more permissive environment, and that pre-malignant cell can now begin to convert to full-blown malignancy. So, this is pretty sobering. This really says that there's been this evolutionary trade-off between this remarkable protection we have from cancer and these phenotypes that we recognize as aging. So of course this is not a very satisfying scenario, and as I said, what we really want to know is whether there's some way we can intervene, and if we can, how can we do it? And we're obviously going to have to do it intelligently. So of course, when biologists are confronted with this kind of dilemma, the first thing we do is we go back to first principles and we ask, what's the mechanism? If we understood the mechanism, we would understand whether we have a chance to intervene. So obviously, today I'm optimistic, so I think we do. And I'll show you 3 very short stories on 3 pathways that we've identified that drive the secretory phenotype and offer the possibility for intervention. And I'm going to, actually 2 of them have already been published, so I'll go through them very quickly. One of them has not been published, but I'll give you the details about that one when I talk tomorrow. So let me start with the first pathway, which is the DNA damage response. So one of the things we noticed is that I told you there are many, many ways to induce cells to become senescent. A few of them, not many of them, but a few of them will cause a cell to acquire many of the characteristics of a senescent cell, but they don't secrete inflammatory cytokines. So these are things like if we overexpress the p16 tumor suppressor, cells will acquire many of the markers of senescence, but they never secrete. And what we realized is that every time we got a phenotype of senescence plus secretion, there was some form of genomic or epigenomic damage. And that led us to hypothesize that the primary driver or a primary driver of this phenotype might be the DNA damage response. So you've already seen a diagram of the mammalian DNA damage response. This is overly simplified, but basically, and I think you'll hear more about this from Vera, but basically what happens is when DNA is damaged, a bunch of kinases get activated, a bunch of adapter proteins amplify that signal, downstream kinases get activated, and in the end, most of that signaling talks to p53, the gatekeeper tumor suppressor. And p53 is largely the decider. It will decide whether the damage should be repaired or whether the cells should undergo senescence or apoptosis. So one prediction we made is that the secretory phenotype should be under the control of p53. That actually turns out to be completely wrong. I bet a bottle of wine on that, and I lost. So here's the data. This is now another array. So these are cells that have senesced with wild-type p53, and these are cells in which we've inactivated p53 by a variety of means. And if you can see these data, if anything, the secretory phenotype is worse. It's higher in amplitude, and actually it develops faster. So one interesting thing about the secretory phenotype, if we induce it with radiation, it takes several days, 4 or 5 days, before the cells begin to secrete. And in the absence of p53, this is accelerated, very much accelerated. And so this is really puts, and p53 is a remarkable protein, but this argues it's even more remarkable because in addition to having these cell-autonomous tumor suppressor functions, inducing apoptosis or inducing senescence, this protein also has the cell non-autonomous activity of trying to restrain this inflammation that's driven by the damage response. But okay, this really begs the question. If it's not acting, if the SASP is not developed through p53, it must somehow be developed by something that's upstream of p53. And Francis Rodier pioneered this work when he was in my lab, and he simply started at the top and started knocking out different DNA damage or DNA damage response proteins. So he started at one of those kinases, ATM. We have very good hairpins that will knock down the expression of this protein in cells. And whether he knocks down ATM activity before he induces senescence, in this case by radiation, or even after he induces senescence, so the cells are fully secreting, he can completely kill, in this case, IL-6 secretion. Now, Francis went on and he did a full array, full antibody array. ATM does not drive the entire secretory phenotype, but it does drive the inflammatory arm. So it's mostly regulating those inflammatory cytokines. So this is sort of good news because there are drugs, they're being used now in the clinic, mostly as anti-cancer agents, that act against this kinase, ATM, as well as this kinase, CHIK2. So Francis was able to show that all three of these proteins participate in driving this inflammation. And the idea is that we might be able to develop drugs that selectively silence these proteins and maybe can selectively silence this inflammation that's driven by senescent cells. Now, although I think this is a great proof of principle, I don't think this is very practical. You need your DNA damage response to ward off and to respond properly to any types of spontaneous damage. So again, this is more proof of principle than promise. But there's a second pathway that was discovered by Adam Freund when he was in my lab. And what Adam discovered is that there's a parallel pathway that works in concert with the DNA damage response that is driven by this stress-activated kinase called p38 MAP kinase. So what he could show is that a p38 MAP kinase is activated in senescent cells, and if he inhibits it, in this case with a chemical inhibitor, he can reduce the secretion, and in this case, 3 different cytokines. p38, it turns out, also drives the transcription activity of this, the activity of this transcription factor, NF-kappa B, which again is suppressed by suppressing p38 activity. And in fact, if you do a Venn diagram of all the cytokines that comprise the secretory phenotype, so that's in purple, you can show that a substantial number is under the control of p38, and a substantial number is under the control of NF-kappa B. He did this by knocking down one of the subunits of NF-kappa B, and there's a lot of overlap. And so this pathway then offers the promise, again, of suppressing inflammation because there are inhibitors in the clinic that are targeted against p38 as well as inhibitors that target NF-kappa B. So this is another possibility. Again, not too practical in my opinion because you also need these activities to mount an acute inflammatory response, which is needed for any kind of acute infection or injury. But the third pathway, which I will tell you about more in detail tomorrow, is actually much more promising. And I think you're going to hear about this from Misha somewhere in the audience tomorrow as well. So as many of you know, and you've heard from Bob earlier, there's been a revolution in aging research in the sense that we now know that there are pathways that drive aging. And if you dampen those pathways, you can postpone aging, delay aging, and improve longevity. And one of those pathways is this ancient pathway that's governed by this kinase called TOR. So TOR stands for target of rapamycin. And what is exciting about rapamycin is that it's been in clinical use for decades in people. It has been called an immunosuppressant because its primary use has been to suppress rejection of organs that are transplanted into people. In fact, it's really not an immunosuppressant. Cyclosporin is the immunosuppressant that prevents organ transplant rejection. What rapamycin did was enable clinicians to lower the dose of cyclosporin so that patients could tolerate the immunosuppressant better. What is particularly exciting about rapamycin is, as you heard from Vladimir earlier, is that this drug has also been shown to extend lifespan in mice. So with a great deal of prodding from my colleague at the Butt, Pankaj Kapahi, and also work that is done primarily by Remy Laberge and Art Orhalo, we've been able to show that rapamycin specifically inhibits the inflammatory arm of the secretory phenotype. I'll show you the data about that tomorrow. But it does it in a really interesting way. And the way it does it is by breaking a positive feedback loop that we discovered over the last couple of years. So in trying to understand the secretory phenotype, what we realized is that there's an initiating cytokine that is activated, and that cytokine is IL-1 alpha. Very unusual cytokine. It's not secreted, it stays in the membrane, and it binds its receptor within the membrane in what's been called a juxtacrine matter. When IL-1 alpha binds its receptor, it activates NF-kappa B. NF-kappa B, in turn, is a very potent inducer of IL-1 alpha, so you make more IL-1 alpha, which then drives more NF-kappa B activity. And finally, you get enough NF-kappa B activity to start driving the transcription of IL-6, IL-8, and all of those other inflammatory cytokines. So this is a positive feedback loop, and parenthetically, there's this microRNA that then comes up and tries to tune this thing down. And what we found is that what rapamycin really does is it breaks this loop. And by breaking the loop, I'll show you data tomorrow, it has long-term benefits, which means this is a drug you probably don't have to take every day. So we can show that at least for 6 weeks, there's a benefit to taking rapamycin with just pulsing cells with the drug for just a few days. So we would like to propose that this may be a new paradigm in thinking about pharmacological interventions into age-related degenerative pathology or hyperplastic pathology that is developing compounds which can play on these feedback loops and therefore are not needed to take every day. And the reason why you don't want to, I know pharmaceutical companies hate this, but the reason why you don't want to take a pill every day is because there simply is no such thing as a completely safe drug. All drugs have side effects. So by minimizing exposure, you can hopefully maximize benefits. So what I've told you today is that we think that senescent cells drive aging and age-related pathology, including cancer, by virtue of driving inflammation. And we've identified a number of pathways that we think are responsible for this senescent-driven inflammation. I'm sure there are others that have yet to be discovered. But these at least give us hopes of handles of ways that we might intervene. We're going to have to be intelligent about how we intervene, though, because again, the senescence response has both a positive effect of preventing cancer as well as a negative effect of driving age-related inflammation. And tomorrow, I'll show you one more piece of unpublished data which suggests there's yet another beneficial effect of senescence, which is again why we're going to have to be intelligent, which is that it promotes tissue repair and regeneration. So we now have to view this complex phenotype as having both positive and negative effects. And when we think about intervening, we're going to have to take into consideration all of the complications of this phenotype. And I'll just stop here again by thanking all the people who worked on the work that I've told you about so far, past lab members, and a great group of outside collaborators. So thank you. Do you want this one back?
No.
Oh, okay.
Yeah, I have a tough question. If the senescent cells produced IL-1, IL-6, these inflammatory secretion enzymes, but why the immune system will not remove these cells? Because
I'm sorry, what was the question? Why?
Why the immune system will not remove senescent cells from the tissues because it secretes the inflammatory cytokines?
Yes. So it's a great question, and the answer is that the immune system does eliminate these cells. So Scott Lowe has, I think, 2 papers now where he clearly shows that senescent cells are targeted for selective killing primarily by the innate immune system. Nonetheless, these cells accumulate with age, and they also accumulate at sites of age-related pathology. So, and in those pathologies, you do see infiltration of the innate immune system. So why are these cells still accumulating, despite the fact that the immune system is clearly clearing them? So the short answer, we think, or at least part of the short answer, we think, is that among this secretory phenotype are a number of matrix metalloproteinases. And matrix metalloproteinases are known to be able to cleave those surface receptors that enable the innate immune system, particularly natural killer cells, to recognize the damaged senescent cell. The proteases can also cleave, so the senescent cells also express cell surface ligands for natural killer cells and other innate immune cells, and the proteases can cleave those as well. So what happens is around those cells that have very high levels of protease secretion, they're creating an immune privileged cage around themselves because they now have soluble ligands, soluble receptors. The soluble ligands, if there are any intact receptors on the NK cells, causes downregulation of the receptors, and so now you have NK cells that are non-functional because they're not expressing the receptors. So we think that's the way the cells get around the immune system, and it's this balance. Sometimes the immune cells get there on time, sometimes they don't. So we think this is how they both talk to the immune system and evade the immune system.
Thank you. So I'm João Pedro Magalhães here.
Where?
I'll get up.
Oh, yes, sorry, yes.
No, I'm actually here, but that's okay. Hi.
Oh, okay.
Thank you for a wonderful talk, by the way.
Thank you.
You mentioned in your talk this idea of a trade-off between cancer and aging, an evolutionary trade-off. But if you look at long-lived species like humans, elephants, naked mole rats, whales, they tend to have low cancer incidence and low
Well, I
cancer
wouldn't say humans have low cancer rates. It's the second leading cause of death.
Well, they do compare to fast-aging animals like mice and rats.
Well, actually, so mice in the wild, I mean, wild-derived mice, Steve Austad has done a fair amount of work of taking wild-derived mice into the laboratory. They have much lower cancer rates than the inbred strains we tend to use in the laboratory, even though so-called outbred strains have gone through severe breeding bottlenecks. So I think probably humans are average for most mammals, but there are some remarkable exceptions, like the naked mole rat, which I don't know if Vera's going to talk about them. Anyway, there are exceptional animals that seem to have very, very low rates of cancer. But for the most part, most mammals get cancer, and if they don't die of it, they die with it.
Judy, over here. There was a really nice talk, and I almost feel like I'm quibbling to raise this point, but since someone else raised it, I'll just elaborate a little bit, which is that I love everything you said, except that I don't see how the aging could have evolved as a response to cancer since it precedes cancer in the lower metazoa, such as C. elegans and Drosophila. I know there's something that passes for tumors in Drosophila, but it's not really.
Say that again. What did you say? What evolved?
I said that organisms that have no cancer,
yes,
that are post-mitotic,
yes,
still age. So
so I'm not arguing this is the only cause of aging.
Okay, good. Okay.
Never.
So, okay.
And in fact, I would argue that post-mitotic cells, like neurons, age by a different mechanism. Right. Okay.
And having said that, I have to comment that the same pathways you're talking about are among those that are suppressed in the very long-lived worms and have been implicated by others as well in aging of C. elegans. So it's clear that we're approaching a unified field theory of aging, if you will. I've got one other question, though, that really does puzzle me, because Michael Lisanti has a cancer host environment model that parallels yours but is different than yours, in that his involves nutrients and mitophagy, in that the cancer cells use hydrogen peroxide to induce the release of ketones from autophagic fibroblasts that are co-cultured with them. Yeah. So can you reconcile these two?
Yeah, I don't think there's a conflict at all. So number one, cancer, so once a cancer is established, it's now independent of the microenvironment. Once you have a truly metastatic cancer, that cell is no longer responding to it's much less responding to its microenvironment than it's sculpting its microenvironment, right? And that's often why it kills. So I think, my view of what Michael's model is really what those late-stage tumors are doing in order to survive, and that's the lethal step in cancer. And what I'm talking about is more about cancer establishment and progression. Oh, I'm sorry. I think it's up to you.
Faustov Rudolf, Computing Center, Russian Academy of Sciences. I'm not a biologist, so my question is the result of my ignorance. But I heard that cancer is caused by retroviruses. Where is their place in your model? It can be. It's not very common. It doesn't happen very often. I have heard that from famous Professor Gallo. I was listening to his lecture once, and he told me that it's the same. The AIDS and that cancer, they both are caused by retroviruses. So let me... Perhaps you know the name of Zilber, Professor Zilber. Yeah. So let me say something that's interesting about HIV. So it's true, HIV is a retrovirus, and it does cause, I think it's generally accepted that it mostly causes a very unusual tumor called Kaposi sarcoma. In fact, patients with HIV/AIDS do not get the major types of cancer. They don't get breast, colon, whatever. Except now, 20 years after long-term antiretroviral therapies, these patients are showing up with age-related cancers of the type that we see in the clinic. So there are two possibilities. One is that the virus does cause the cancer. Actually, I don't think that's the case. I don't think there's any evidence that that's the case. But the other is that either the inflammation driven by the virus or the antiretroviral drugs themselves, which cause things like mitochondrial mutations,
And laminate misprocessing may be driving those late life cancers. So I think there's, we can't say that there's no retrovirus that causes cancers. There are some, but I think most cancers are caused by spontaneous mutations in a degraded tissue environment.